Microtubules. Hello friends, today's topic is microtubules. Microtubules are found in the cytoplasm of all types of eukaryotic cells within the absence, such as in human erythrocytes. They are tiny, hollow, bead-like tubular structures that help cells to maintain their shape. They are microscopic hollow tubes found inside the cells that also provide motor functions for the cell. Structure of microtubules. They are long fibers of indefinite length, about 24 nm in diameter. In cross section, each microtubule appears to have a dense wall of 6 nm thickness and light or hollow center. In cross section, the wall of microtubule is made up of 13 globular subunits called protofilaments, about 4 to 5 nm in diameter. Chemically, they are composed of two kinds of protein subunits, alpha tubulin and beta tubulin, each of MW 55,000 daltons. The wall of microtubule is made up of a helical array of repeating alpha and beta tubulin subunits. Assembly studies have indicated that the structural unit is an alpha-beta dimer of 8 nm length. Thus, in each microtubule, there are 13 protofilaments, each composed of alpha-beta dimers that run parallel to the long axis of the tubule. The repeating unit is an alpha-beta heterodimer which is arranged head to tail within the microtubule, that is alpha-beta. Okay. Thus, all microtubules have a defined polarity. Their two ends are not structurally equivalent. Assembly. Microtubules undergo reversible assembly, disassembly, polymerization, depolymerization, depending on the need of the cell or organelles. They polymerize is regulated by certain microtubule associated proteins. The assembly of microtubules involves preferential addition of subunits to one end of the tubule called A end, okay, and the other end of the tubule is D end. Such an assembly involves the hydrolysis of GTP to GDP. Thus, the assembly of the tubulin in the formation of microtubules is a specifically oriented and programmed process. Centrioles, basal bodies and centromeres of chromosomes are the site of orientation for the assembly. Calcium and calmodulin and acidic protein having 4 regulation factors in the 2 polymerization of tubulin. Okay. Then the microtubules may work alone or join with other proteins to form more complex structures. Cell organelles derived from special assemblies of microtubules include cilia and flagella. Then second one is basal bodies and centrioles. Functions of microtubules. They move vesicles, granules, organelles like mitochondria and chromosomes via special attachment proteins. Okay. Along with microfilaments and intermediate filaments, they form the cytoskeleton of the cell as well as participate in a variety of motor functions for the cell. As part of the cytoskeleton of the cell, microtubules contribute to giving shape to the cells and cellular membranes. Second one is cell movement, which include a contraction in muscle cells and more. Then third one is transportation of specific organelles within the cell via microtubule roadways and or conveyor belts. Then the fourth one is mitosis and meiosis movement of chromosomes during the cell division and creation of the mitotic spindle.